Our chosen topic is about the applications of molecular phylogenetics in autoimmune diseases. Basically, phylogenetic is a study of phylogeny based on evolutionary relationships which happen to be a basis for cladistic analysis while making use of molecular sequence as well as protein sequence data to verify whether a trait is homologous or analogous. Phylogenetic analysis consists of two major components, which are phylogeny inference and analyzing change. By using stratified data analysis, phylogenetics has two common analytic methods that are character-based and distance-based methods. Apart from that, the analysis uses taxon sampling, which uses four different tree construction models, which are neighbor joining, minimum evolution, maximum likelihood, and maximum parsimony. For your information, Bayesian phylogenetic inference is one of the most popular approaches for tree reconstruction. This method helps to infer phylogeneous and phenotypic trait evolution, uncertainty, analyze molecular dating, and dynamics of specific extinction. Examples of works used are MyBayes and PhyloBayes as stated above. Moving on to the next part.
Autoimmunity, a captivating realm of scientific inquiry, unravels the enigmatic workings of our immune system. It explores the phenomenon where our body's defense mechanism mistakenly attacks our healthy cells and tissues. This intricate process entails a breakdown in self-tolerance, leading to the development of diverse autoimmune diseases. Autoimmune diseases are conditions where the immune system, originally designed to shield us from external threats like bacteria and viruses, loses its ability to distinguish between our cells and foreign cells. Consequently, the body mistakenly attacks our healthy cells and tissues, causing inflammation and damage across various organs. Rheumatoid arthritis, lupus, and multiple sclerosis are just a few examples of the 80+ identified autoimmune diseases, each affecting specific organs or systems. These conditions manifest with a spectrum of symptoms ranging from chronic to episodic. While the precise causes of autoimmune diseases remain elusive, a combination of genetic, environmental, and hormonal factors may play a role. Regrettably, most autoimmune diseases lack a cure, necessitating lifelong management to alleviate symptoms. Hence, understanding the underlying mechanisms behind autoimmunity is crucial for developing effective treatments and interventions. So, let us embark on a journey together to explore the captivating realm of autoimmune diseases.
In general, autoimmune diseases are a group of roughly 80 polygenic diseases characterized by a variant immune response, where healthy tissue, cell, and organs are attacked. This is caused by the failure of immune system to respond appropriately to self-antigen. Environmental factor, especially genetic factor, contribute significantly to autoimmune disease. Hence, database and sequencing gene can help in analyzing disease-causing gene to understand the causes and may be able to generate the solution. The following are some examples of database and sequencing technology used in autoimmune disease. The first one will be genome-wide association study. It can identify several risk loci. However, it has its own limitation too, which is it only able to account for a small proportion of estimated heritability. This may be due to it only examining common single nucleotide variant. And the next one will be UMI sequencing. This method commonly used to sequence heterogeneous cell population. It can be done by attaching an UMI to individual input DNA molecule. After sequencing, the software will deconvolute the UMI and the UMI will be collected for analysis. Each UMI represents each input DNA molecule. This method combined with single-cell omic technology to analyze large number of data. Moving on, the next one is long-read sequencing. The purpose of this method is to resolve large complex genetic variants such as repeat expansion, copy number, and the structural variation, and also for sequencing the TCR or BCR and HLA region. For the information, TCR is referred to the T cell receptor, BCR is B cell receptor, while HLA is human leukocyte antigen region. In autoimmune disease, it may be used for pseudogene determination as well. It is a study of a diagnostic application that functions to sequence 3 autoimmune disease genes while bypassing the IKB KGP1 pseudogene. And lastly will be short-read DNA-based method. Here, I only take gene panels, exome and whole genome to make the comparison, and as an example of the sequencing option. And through this comparison, we can say that in terms of function, we can conclude that the whole genome is the best option.
I'll be explaining these applications and technology used in molecular phylogenetics for autoimmune diseases, along with the concepts and technologies involved.
So firstly, genetic susceptibility analysis. So what does this mean? Genetic susceptibility means being more likely to develop a certain disease due to a mix of your genes and the environment. So to explore this, we check the evolution of genes linked to autoimmune diseases. So for this study, we use the next generation sequencing technologies, NGS. So it helps us quickly and affordably analyze the order of DNA or RNA building blocks. So this technology is crucial for understanding the genetic roots of autoimmune diseases. And for example, Illumina or Ion Torrent for gene sequencing and bioinformatic tools for variant calling and analysis. Secondly, the identification of diseases-associated genes. So finding disease-associated genes means figuring out which specific genes or genetic changes are connected to the development or higher risk of a particular illness. So in this case of autoimmune diseases, it's about uncovering the genetic factors that make someone more prone to these conditions. So to do this, we analyzed how genes have evolved, looking for patterns related to autoimmune diseases. We use computational tools like BLAST for comparing genomes to spot genetic differences linked to these diseases. So we also use the tree building algorithms like RAXML and PhyML to study how immune system genes have changed over time. So thirdly, phylogenetic analysis of immune system genes. Studying immune system genes through phylogenetic analysis means looking at how these genes have evolved across different species. So when we use this approach for autoimmune diseases, it helps us understand the genetic reasons behind the immune responses going haywire and causing autoimmunity. So tools like MEGA, PAUP, and PhyloBase are important for this kind of analysis, because by using these tools in genetic studies, we can learn how the immune system has changed over time and how genetic differences play a role in causing autoimmune diseases.
Alright, now let us go through the research examples about the HLA-G polymorphisms are associated with non-segmental vitiligo among Brazilians. Before that, I would like to explain one of the applications of molecular phylogenetic that scientists usually use, which is called as single-cell RNA sequencing. So basically, this technique, it allows researchers to examine the gene expressions of individual cells. So this technique is to provide the insight into the immune system complexity and the mechanisms of autoimmune diseases. So what is vitiligo? Basically, vitiligo is a skin condition with white patches due to the loss of melanocytes. So the current treatments of it is to slow skin depigmentation, or we can also repigment the affected areas. So the autoimmune feature of this disease is that it is suggested based on the occurrence of other autoimmune diseases in vitiligo patients and relatives, and involvement of immune-related genes. So basically, the study of vitiligo, it used massive parallel sequencing. Basically, the study of HLA-G and vitiligo in a Brazilian population is to use advanced sequencing to study the variations and combinations in the HLA gene. So it includes all regions and exons in vitiligo patients and healthy individuals from the Brazils. So basically, the purpose of this study is to determine if changes in the HLA-G gene could affect the likelihood of developing vitiligo. Now, let us go through to the HLA-G and its role in vitiligo. So basically, the functions of HLA-G is to inhibit the natural killer of cells and cytotoxic T lymphocytes. How it works, it is by binding to leukocytes receptor on the cell surface, which includes ILT2, ILT4, CD160, and KIR2DL4. So basically, this includes inhibitory tyrosine motifs. Next, what is the correlations with vitiligo? So HLA-G expression has been found to be negatively correlated with vitiligo among Tunisians. So this suggests that HLA-G may influence the development of vitiligo. So in conclusion, HLA-G is seen as a potential gene which causes vitiligo. Next, the HLA-A02 alleles. These molecules can trigger immune responses by presenting many antigens from melanocytes proteins. However, their role in causing vitiligo is still being debated. Lastly, the HLA-G 01010101 UTR1 allele. So this allele codes for a well-known immune modulating molecule and is linked to different expressions profiles.
Welcome to our exploration inspired by the article Arthritis and Prolactin, a phylogenetic viewpoint. Arthritis marked by joint inflammation is not exclusive to humans. Fossil evidence traces its presence in dinosaurs, mammals, and humans. Today, we will delve into the evolutionary nuances of spondyloarthritis and rheumatoid arthritis. Our journey starts with spondyloarthritis, the most prevalent form in vertebrates, found in diverse species from amphibians to mammals like primates. Its deep-rooted prevalence raises questions about the interplay of genetics and the environment in shaping arthritis. Notably, gout and osteoarthritis are less common in the wild, gaining prominence in the captivity. This suggests a complex relationship between genetics and the environment. The widespread occurrence of spondyloarthritis across vertebrate taxa suggests its evolutionary significance. This prompts us to consider its potential adaptive nature over the course of evolution, playing a crucial role in the health and adaptation of various species. In contrast, rheumatoid arthritis is uniquely human. Emerging 5,000 years ago, it may have originated from spondyloarthritis. Its exclusive occurrence in humans invites us to explore the distinct immunological landscape that sets us apart and the evolutionary forces shaping this specific form of arthritis. Despite its seemingly debilitating nature, arthritis might have served an evolutionary purpose. The thrifty adaptation hypothesis suggests that the pain associated with arthritis could compel organisms to conserve energy during adversity such as starvation, stress, pregnancy, lactation, and old age. On the molecular level, we explore the role of prolactin PRL, a hormone with evolutionary conservation. PRL found in various vertebrates has high big effect on stress, immune interaction, hinting at its potential role in the adaptive regulation of arthritis development. In summary, arthritis as an autoimmune disease exhibits intriguing patterns across the evolutionary tree, while spondyloarthritis emphasizes its importance in adaptation across vertebrates. Rheumatoid arthritis stands as a distinctive human condition with a historical connection to its broader vertebrate counterpart. Understanding this evolutionary aspect sheds light on autoimmune disease diverse nature, offering valuable insight into their origins and impact on different species over time. Thank you.